Next chapter, the cell. Now, we have learned about the characteristics of living organisms. Now, we need to go down further and learn what constitutes these living organisms. Living organisms like human beings are made of organ systems such as the digestive system. Each of these organ systems comprise individual organs. Here, organs such as liver, stomach, and the small and large intestines are part of the digestive system. All these organs are made up of tissues such as the epithelial tissues and the smooth muscle tissues. Tissues are made up of cells. The cells contain organelles and molecules such as DNA. A cell is the smallest unit of life and the building block of all living organisms. It is defined as the basic functional unit of an organism capable of growth, development, reproduction, and all other characteristics of a living organism. Understanding what makes up a cell and how the cell works is fundamental to all biological sciences. In this chapter, we shall also appreciate the similarities between cell types between and within organisms. But as you can already imagine, a cell is very small in size, and we need specialized instruments to be able to visualize it. The magnifying lenses. When you want to visualize something small, you can bring your eyes closer. But if you bring your eyes too close, the object looks blurred. That's because the eye cannot focus on really close things. A magnifying lens can bring the object into fine focus and make it look bigger. This is called the image. The ratio of the image size to the object size is called magnification. Therefore, mathematically, magnification is equal to image size over the object size. The magnification of a magnifying lens is still not sufficient to visualize cells. By combining several lenses, where the enlarged image of one lens becomes the object for another, it is possible to obtain a final image that is magnified several hundred folds. Using this concept, scientists developed the compound microscope, or better known as light microscope. This is a light microscope. It is called a light microscope because it uses light to illuminate objects under view. Let us first study the parts of a microscope. The eyepiece or the ocular is the part of the microscope closest to your eye, through which you look. It contains the ocular lens, which magnifies the image produced by the objective lens. It is usually labeled with its magnification, like 5x or 10x, meaning it enlarges 5 times or 10 times. The body tube holds the eyepiece in position as well as the revolving nosepiece. It may be straight up or slanted. The arm is the curved metal piece that holds the body tube in place as well as the stage. The revolving nosepiece holds the objective lenses in place and allows change from one objective lens to another. The objective lenses are closest to the sample under view. They bring the object into fine focus and magnifies it. The larger one holds stronger lenses, the high power lenses. The shorter one holds the weaker lenses, the low power lenses. Like the eyepiece, objectives also come in various powers, like 4x, 5x, or 40x, or 45x, and 100x. Some microscopes only have 1 or 2 objectives. The stage is the flat surface where the specimen is placed. The stage clips hold the slide in place on the stage. Beneath the stage is the condenser, which collects and concentrates light on the object on the stage. The iris diaphragm regulates the amount of light passing through the condenser to illuminate the specimen. The coarse adjustment knob is the larger knob, which raises and lowers the body tube for long distances to bring the image into fine focus. The fine adjustment knob raises and lowers the body tube for short distances to bring the image into finer focus. The mirror, located beneath the stage diaphragm, reflects light through the condenser to illuminate the specimen. The base of the microscope is the heavy bottom part that provides firm and steady support. The substage adjustment knob is used to move the condenser up and down. When using the microscope, hold the microscope firmly with both hands, one hand under the base and the other on the arm. Place it on a table away from the edge. Move it to a position in which you can look comfortably through the eyepiece. Ensure the low-power objective is in place over the hole in the stage. Use the coarse adjustment knob to raise the body tube so that the objective is about 2 centimeters above the stage. Put the slide on the stage so that the part you want to look is over the hole and under the lens. Use the clips to hold the slide in place. Look through the eyepiece. You should see a circle of light against a black background. Adjust the mirror to make the circle as bright and even as possible. If you can't see any light, your objective is probably not in line with the ocular tubes. Adjust the revolving nosepiece until you hear it click in place. The mirror has one side flat while the other is concave. It is not possible to say which one is best to use in all cases. You will have to try and see which one gives you a better view. The flat side will provide a sharp image in most cases, while the curved side will provide a brighter image. Never use the microscope in direct sunlight, as the reflected light could damage your eye. If you are using artificial light, put it about 20 centimeters away from the mirror. Adjust the lamp so that the light points towards the mirror under the stage. Look through the eyepiece and use the coarse adjustment knob to find good focus of the image. When the specimen comes into focus, it will become clear. To use the high-power objective, turn the revolving nosepiece to bring it into position over your sample or slide. Now use the fine adjustment knob to find the fine focus of the specimen. Some microscopes have a special type of lens called the oil immersion lens, 100 times. This lens is used by placing a drop of oil on the top of the cover slip and adjusting the position of the objective until it touches the oil. The oil bends light more than air so that more light can enter the microscope. Magnification. The magnification of the object under view is calculated by multiplying the eyepiece magnification by the objective lens magnification. If the eyepiece magnification is times 5 and the objective used is times 40, Then the total magnification will be 5 times 40, giving us 200. So the total magnification is times 200, meaning that the specimen is enlarged 200 folds. The electron microscope. The electron microscope is more powerful than the light microscope. It has more complicated parts. The electron microscope can magnify up to 500,000 times and has a higher resolving power. The high resolving power of the electron microscope enables it to make out as separate things, objects which lie close to one another. This high resolving power makes the electron microscope an important research tool. The electron microscope uses a beam of electrons instead of light to illuminate objects. Now we move to the cell structure. The bodies of organisms are made up of billions of cells. All cells have 3 things in common, no matter what type of cells they are. All cells have cell membranes, which separate the inside of the cell from its environment. Cytoplasm, which is a jelly-like fluid, and DNA, which is the genetic material of the cell. The cell membrane is also called the plasma membrane. The plasma membrane surrounds the cell and separates the interior of the cell from its environment. This semi-permeable membrane is composed of lipid bilayer made up of phospholipids. The plasma membrane is embedded with proteins and also contains cholesterol. Small particles and molecules like oxygen can diffuse through the plasma membrane by moving in between the phospholipid bilayer. The membrane selectivity allows materials in and out of the cell. It is thus said to be selectively permeable or semi-permeable. Organelles simply means little organs. They are the specialized part of a cell with unique roles to perform. We start with the nucleus. This is the control center of the cell. The nucleus contains DNA, or the genetic material. The DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. Inside the nucleus is the nucleolus. This is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have an important job in synthesizing proteins. Outside the nucleus, the ribosomes and the rest of the organelles float in the cytoplasm. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum known as ER. There are 2 types of ER. The rough ER has ribosomes attached to it, and the smooth ER does not have any ribosomes attached to it. The ER is a passageway for transporting materials such as proteins synthesized by the ribosomes. And other materials emerge from the ER in small vesicles and move into the Golgi apparatus, sometimes called the Golgi body. In the Golgi body, the proteins are modified into forms that the cell can use. This includes folding the protein chains into 3-dimensional structures or adding other materials to them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials. In plant cells, the central vacuole stores water and contributes to the osmotic properties of the cell. Back to the animal cell are organelles called lysosomes. These are garbage collectors that take in foreign materials or worn-out organelles. They contain lytic enzymes that break down these materials. The mitochondria is the powerhouse of the cell. It is the site for cellular respiration where ATP is made that provides energy for cellular activities. It's double membrane with an inner membrane folded into cristae. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through the cytoskeleton. This includes the thread-like microfilaments made of proteins and microtubules, which are thin hollow tubes. Some organisms, such as plants that are photoautotrophs, meaning they capture sunlight for energy, have cells with organelles called chloroplast. This is where photosynthesis takes place. It's green because it has a green pigment called chlorophyll. Plant cells also have a cell wall outside their cell membrane that shape, support, and protect the plant cell. Animal cells do not have cell walls. There are many other unique structures that are only present in some cells. In humans, for example, the respiratory tract is lined up with cells that have cilia. These are microscopic hair-like projections that can move in waves. This feature helps them trap any inhaled particles in the air and expel them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that have a flagellum is a sperm cell. We need to perform 2 experiments to prepare temporary slides. In the first experiment, we will prepare a temporary slide of onion cells. For this experiment, we shall need a medium-sized onion, a slide, a cover slip, watch glass, forceps, a knife, needle, distilled water, glycerin, dropper, blotting paper, saffron in solution, a compound microscope, and a brush. Slice the onion into 4 or 5 pieces with the knife. Remove a scale leaf from one of the pieces. Break it gently from the middle and peel a small portion from the delicate epidermis. That is the covering of the inner surface of the scale leaf. Remove the delicate epidermal peel with a forceps and place it in a watch glass containing distilled water. Take another watch glass and place 3 to 4 drops of saffron in solution, or you can use iodine solution. With the help of a brush, transfer the peels to the watch glass containing saffron for staining and wait for 2 to 3 minutes. Now transfer the peels into distilled water to remove the extra stain sticking to the peel. Take a clean glass slide and pour 1 to 2 drops of glycerin. Transfer the stained peels onto the drops of glycerin on the slide with a brush. Flatten the peel with the help of a needle and brush. Now place the cover slip on the slide in such a way that you do not allow air bubbles to enter. Gently press the cover slip with a needle to spread the glycerin evenly. Wipe out any extra material surrounding the slide with a blotting paper. Now examine the slide under the light microscope. You will see a large number of cells lying side by side with distinct cell walls. A large stained nucleus is present in the periphery of the cytoplasm. A prominent vacuole is the center of each cell, surrounded by the cytoplasm. Now take note that we took a thin layer to ensure that it's one cell thick. Organelles in a cell pick the stain to different degrees, and hence the stain helps us in distinguishing the organelles. In conclusion, this is a plant cell because of the presence of the prominent vacuole and also the presence of the cell wall. In the next experiment, we will prepare a temporary slide of human cheek cells. We will require a slide, a cover slip, a toothpick, glycerin, methylene blue solution, a blotting paper, a compound microscope, a dropper, and water. Take a clean slide and in the middle of it, pour a drop of water with the help of a dropper. Take a clean toothpick and use it to scrub the inner wall of your cheeks gently so as to scrub the epithelial tissue. Mix the scrub on the toothpick on the drop of water on the slide. Pour a drop of methylene blue solution on the mixture on the slide and mix it properly. This will stain the epithelial tissue. After 2 to 3 minutes, remove the excess water and methylene blue solution using a blotting paper. Pour a drop of glycerin on the slide content and spread it. Take a clean dry cover slip and place it on the slide. Remove any extra material surrounding the slide with the blotting paper. Now examine the slide under a compound microscope. You will see a large number of flat irregular cells. Each cell has a thin cell membrane. A distinct, deeply stained nucleus is seen in each cell. The space between the nucleus and the cell membrane is filled with the granular material called cytoplasm. There are no intercellular spaces between the cells, no visible cell wall. This suggests that the specimen is that of animal cells because there is no prominent vacuole visible. Next, cell specialization. Cells that are differentiated or modified to perform specific functions are said to be specialized. In unicellular organisms, a single cell is capable of performing all the biological functions needed by the organism for survival. In multicellular organisms, cells have to be specialized because of the complex nature of the organism. Examples of specialized cells in animals include neurons found in the nervous system. They are specialized for transmission of nerve impulses from different parts of the body to the brain and also from the brain to the receptor organs. Smooth muscle cells are responsible for involuntary movement of our bodies. They form the walls of blood vessels, digestive tract, urinary tract, respiratory tract, and so on. Red blood cells are specialized for transport of oxygen from the lungs to other cells of the body. They also carry carbon dioxide from cells to the lungs for exhalation. They are biconcave in shape and lack a nucleus to give room for packaging of hemoglobin, the oxygen transport molecules. Sperm cells are the male reproductive cells. They fuse and fertilize the mature female cells, ova. They have a tail or flagellum which aids in their movement. Specialized plant cells include guard cells. They are specialized epithelial cells found on the leaves and stems of some herbaceous plants. A pair of guard cells controls the opening and closing of stomata to allow exchange of materials between the plant and the outside environment. Root hair cells are specialized epithelial cells present on the roots. They help in the uptake of water and minerals for the plant. They have a hair-like outgrowth on the surface, which increases the surface area for uptake of water and mineral salts from the soil. The vacuole is large and occupying almost all the space in the cell, leaving only a thin layer of cytoplasm. The vacuole is filled with a solution that maintains its osmotic properties and allows for absorption of materials. Next, we look at tissues. And we say that a tissue is a group of cells of the same type that are grouped together and perform the same function. Animal tissues are grouped into 4 basic types: epithelial, nervous, muscles, and connective tissues. Epithelial tissue covers and protects the external and internal surfaces of the animal body. Epithelial tissue is divided into simple squamous, stratified squamous, columnar, and cuboidal epithelium based on the shape and function of the cell. The simple squamous is found on the linings of the alveoli and allows for transport of small molecules across the membrane. It is extremely thin, flat, and single-layered. The stratified squamous epithelium protects areas subject to friction and are found on the skin. It is multi-layered. Columnar epithelium acts as an impermeable barrier against any bacteria that could be ingested but are permeable to any necessary ions. It's tall and shaped like a column. Cuboidal epithelium aids in the absorption and provides mechanical support. It forms the lining of the kidney tubules and it's shaped like a cube. The next tissue is the nervous tissue, which consists of a nerve cell or neuron that has a cell body containing nucleus. The cell body extends and forms specialized cell parts called dendrites and axon. These are the cells that help you retract your hand when you touch a sharp object. The third tissue type is the muscle tissue that is responsible for movement within our bodies. The 3 types of muscle tissue are skeletal, smooth, and cardiac muscles. The skeletal muscles help us in locomotion and other voluntary body movements. It is made of long fibers with alternate light and dark bands or striations. So skeletal muscles are also called striated muscles. Smooth muscle cells provide involuntary movements such as contraction of walls. They line the walls of hollow structures of the body, like intestines and blood vessels. They are spindle-shaped, flat, and broader in the middle. The cardiac or heart muscles are involuntary muscle cells that contract and relax throughout life. They are cylindrical, branched, and with single nucleus. Finally is the connective tissue. This binds and supports body parts and transport materials. Connective tissues are subdivided into blood, bone, ligament, cartilage, areola, and adipose tissue. The blood contains red blood cells, white blood cells, and platelets that transport gases and other materials to different parts of the body. Blood is a fluid matrix. Bone cells secrete calcium that hardens bones and support the main organs of the body. Bone cells are solid. Ligaments function as the connection between bones. They are fibrous and stretchy. cartilage protects the bones by preventing them from rubbing against each other. It is a tough but flexible tissue. Areola fills the space between organs. It's jelly-like with both elastic and inelastic fibers. Adipose tissue stores fat molecules. And now plant tissues. Plants are composed of three major organs: roots, stem, and leaves. These organs are made up of various tissues that perform different functions. Large number of specialized cells form a tissue. Plant tissues are characterized and classified according to their structure and function. The two basic types are meristematic and permanent tissues. The meristematic tissues are responsible for the growth of a plant as they have the ability to divide. Their cells are similar, generally small, enclosed within walls, and are cubical. Depending on the region they are present in, meristematic tissues are classified as apical, lateral, and intercalary. The tissue that helps increase the length of the stem and root is the apical meristem. It is located at the tip of roots and stem. The lateral meristem helps to increase the diameter of the stem. The plant also needs to grow branches. This is facilitated by the intercalary meristem. In grass, these generate the parts removed by the grazing herbivores. It is located at the base of the nodes, internodes, and on leaf bases. For plants to grow, they will need constant supply of materials. It is the permanent tissues that help in the transport, preparation of food, and provision of support for the plant. Permanent tissues are just meristematic tissues that mature and become differentiated to form different types of tissues. Permanent tissues are classified into simple permanent and complex permanent. Simple permanent tissue is called simple tissue because it is composed of similar types of cells that have a common origin and function. Based on the function, they are further classified into parenchyma, collenchyma, and sclerenchyma tissues. Parenchyma are oval-shaped cells within cell walls and have extensive spaces between the cells. They aid in photosynthesis, storage, and also help aquatic plants to float. They are composed of living cells found in roots, stems, leaves, and flowers. The collenchyma tissues are composed of living elongated cells. They provide elasticity, mechanical support, and tensile strength to the plant body. They are found mainly on the cortex of stem and leaves. The sclerenchyma tissue are dead cells having extremely thick cell walls containing lignin. They protect the plant. This tissue is present in stems, leaves, and in the hard covering of seeds and nuts. Now the complex permanent tissue. Complex permanent tissue consists of more than one type of cells. They are classified into xylem and phloem. Xylem is a complex tissue consisting of tracheids, vessels, xylem parenchyma, and xylem fibers. It transports water and minerals from the roots to other parts of the plant. Phloem consists of sieve tubes, companion cells, phloem fibers, and phloem parenchyma, which transport prepared food from leaves to other parts of the plant.